we learned that different strains of HSV1 early in infection have a different relationship with CTCF. This suggests that maybe each strain has sort of a fingerprint of enrichment. Next, we learned in the 293T cells that assembly of CTCF onto the genome appears to be something that some cells are good at, like our neuro-2A cells are a little bit better at it than the 293T cells, and so maybe neuronal cells have a different mechanism of dealing with incoming HSV genetic material than, say, the 293T cells would. Our DNA replication studies suggest that DNA binding protein, like CTCF, might be incompatible with DNA replication from the virus. This is consistent with work studying reactivation of both HSV1 and KSHV, in which reactivation involves the eviction of CTCF from the genome. And this is work done by a number of labs in KSHV, especially the Lieberman lab, and Donna Newman at LSU is responsible for the HSV1 reactivation work that has shown this. We have also seen that CTCF and viral gene expression are complicatedly intertwined, and that knockdown alters the way the virus can express its genes. And if you alter the viral transcription program, you can change how CTCF is enriched on the genome. It also seems like, depending on the multiplicity, CTCF can act either as a positive or negative regulator of transcription, based on the knockdown data. So here's a little bit of a graphical model to sort of summarize this. If you go in with different numbers of genome, the cell, in this case a neuronal cell, has a differential capacity to load CTCF on. When you start to have transcription, we do know that you only get transcription from a limited number of genomes, even if you go in with a high multiplicity. So perhaps those genomes become under-enriched, whereas other genomes that are not being used as template do not. Whereas in a low multiplicity, you don't have a whole lot of genomes to choose from in the cell. And then by the time you get DNA replication, you get gross eviction of CTCF. What about if you go in with a mutant that can't transcribe very well? Well, this pattern doesn't change much between 0 and 3 hours post-infection. And lastly, if you go in with an epithelial cell, you start this race behind because you can't assemble CTCF onto the HSV1 genome. So this might suggest that different kinds of cells occupy a different space on the ability to assemble chromatin factors onto the HSV1 genome. And so on the right here, we would have severely repressed or latency. On the left, we would have very permissive infection. And so neuro-2A cells are a little bit better at silencing the HSV1 genome or assembling chromatin factors onto it. And so they occupy this space here, but they're not good enough at it to push it over the threshold into latency. So far, the only cells that we know that can do that are sensory neurons, but we also know that sensory neurons can also support lytic replication. So there exists the potential for maybe different cells, even at the same cell type, to have a different ability for this. And of course, down here, 293T cells, they're not very good at this at all, and so they don't stand a chance. So, sort of take-home point from this research is that CTCF is part of the ongoing war between the infected cell and the virus, as the cell attempts to suppress the virus, and the virus must then overcome this response. All right, that's all I have.